Measurement of microbial cells. Measurement of microbial cells involves different methods in order to count the microbial cells. We have plate count method or plating method, turbidometry method, and Coulter counting method. Let us discuss about plating method. In plating method, we have to dilute the sample to be counted. We can do it by conducting serial dilution of the sample to be counted. For this, we have to take 1 ml of sample to be counted and add it to test tube containing 9 ml of saline labeled as 10 raised to minus 1. Then from that tube, we have to transfer 1 ml of sample to 10 raised to minus 2 tube. Same thing we have to repeat for all the tubes. Then by pour plate or spread plate method, we can inoculate the sample and incubate it. After incubation, colonies will appear. These colonies can be counted. In 10 raised to minus 1 and 10 raised to minus 2 tube, there are too many colonies and cannot be countable. So in 10 raised to minus 3 tube, you can see a little lesser colonies. The colonies become lesser with the dilution factor. Once you get the colonies, you can count them and multiply with dilution factor. The counted cells can be represented in forms of the unit CFU, that is colony forming units. Next we have Coulter counting method. Coulter counting method is an electronic method. In this method, microbial suspension is forced through small hole or orifice. Here the movement of microbe through orifice impacts electric current that flows through the orifice. In this method, living and dead cells are not distinguished. It is a quick and easy method. It is used for large microorganisms and blood cells counting. In the picture, you can see the device wherein microbial suspension is added through the counting orifice. Cells will be counted in counting orifice, which is attached to electronic detector. Then the counted cells will be displayed in automatic counter display. So this is about Coulter counting method. Next we have turbidometric method. Turbidometric method is a simple method in order to follow the growth of microorganism. It is a spectrophotometer or calorimeter can be used for turbidometric measurements of cell mass. Spectrophotometer is used to determine turbidity, that is cloudiness, by measuring the amount of light that passes through a suspension of cells. If there is more cell, turbidity will be more. Likewise, if turbidity is more, less light passes through the suspension. The culture to be measured must be dense enough to register some turbidity on the instrument. Cultures grown in deeply colored media can be counted, or cultures that contain suspended material other than bacteria also. It must also be recognized that dead as well as living cells contribute to turbidity. Percentage transmission. If fewer cells are present, turbidity will be less and thus will allow more light to pass through. Therefore, percentage transmission is higher when the cell number is lower. Percentage transmission will be higher when the cell number is lower. So, absorbance is the opposite of percentage transmission. More light is absorbed when more cells are present and thus absorbance goes up as turbidity goes up. That is, cell number increases with the increase in turbidity. This is the principle behind turbidometric method. First, the uninoculated blank sample is kept and set the reading to blank. Then the sample that containing bacterial suspension is kept in order to take the reading. The readings of all the tubes are recorded in regular intervals of time. Then the readings will be plotted by taking time on x-axis and absorbance on y-axis, and the readings or graph will give you a curve. That curve will call it as growth curve, that you have already read it. So this is about the turbidometric method. These are the brief explanation of all types of counting methods.